So it's saying we are live.
Okay.
It might take some time to come over there, but okay. So will it not start before 12?
Maybe not.
Because I actually set the time to 12. So.
Okay, so maybe it won't. So we'll just have it broadcasting there, and then we can see at 12 if it pops up. Okay, sweet.
Books.
we move this? Yeah.
You can see here. This is the angle that you'll be getting.
So maybe walking. No, we're not. Don't worry about that.
There's supposed to be a handheld mic, but I'll get it from the guy back there. Don't you all look great? Thank you very much for coming to this, the last installment of the Borlaug lecture series for this semester. I'm Dr. Elsa Morano, Director of the Borlaug Institute, and it's just wonderful to see everybody here because we have a really terrific speaker. I think you will be delighted with his presentation this afternoon. So what I wanted to do is tell you that our Borlaug seminar series, which has been ongoing, of course, this semester, we've had an incredible roster of speakers, people like the Director General of the ICA and the Deputy Director of the Institute, International Livestock Research Institute, to the President of El Zamorano University in Honduras, the President of the National Chimsing University, and this goes on, the President of the Ajo Foundation, but we have an extra special treat for you this afternoon. And to introduce our speaker, I'd like to welcome to the front our Associate Vice Chancellor for Business and Strategic Operations and also Associate Dean of the College of Agriculture and Life Sciences, Dr. Bill Dukes.
Thank you, Elsa. Good afternoon. We're glad you're here. There are a few seats up front, so don't be bashful. Those of you in the back, come on up. I know Robb's interested in seeing your faces up close and personal. Robb Fraley is the Executive Vice President and Chief Scientific Officer for Monsanto, where he has worked for more than 30 years and oversees the company's global technology division. He grew up on a small farm in Illinois and earned a PhD degree in microbiology and biochemistry from the U of I, and did a postdoc at UC San Francisco. He's the recipient of the National Academy of Sciences Award for Industrial Application of Science. He's a fellow of AAAS and a recipient of the World Food Prize, which, as you know, was founded by Dr. Borlaug for, quote, breakthrough achievements in founding, developing, and applying modern agricultural biotechnology. I suspect we'll hear a little bit of that today, given the title above my right shoulder. We here at A&M are greatly appreciative of our long-term and synergistic relationship we have with Monsanto, including their support of the Borlaug Monsanto Chair in International Crop Improvement, the Borlaug Beachville International Scholars Program, and for graduate students. The title of this talk is over my right shoulder. I will not repeat it. We look forward to a great lecture and a vibrant discussion with our students and our faculty. Please join me in giving a warm Aggie welcome to Dr. Robb Fraley.
Great. Great to be back here. Everybody doing okay? I'm going to get ready for this. I'm good. Thanks. Had a busy morning this morning. We were out with the beekeepers this morning. That was a lot of fun. I appreciate it. We had a great time. Learned a lot. Had a chance to meet with several of the students, both some of the Beachville Borlaug scholars, some other students. Had a chance to meet with 3 or 4 really exciting entrepreneurs doing really cool stuff. Right? Let's get started. So, great visit. And like I said, great to be back. I've been on a visit here to, as I always do, to learn what's going on and what's exciting. So we've done a little bit of that. I've had a chance to talk about some of the things that we're really passionate about. One of the things I'll talk about at the end of my talk is, we all have a connection to science. I've been trained as a scientist all my life, but I want to emphasize how important it is not only to do the science, but to talk about it and reach out and talk to other people about that science across society. So we'll have a chat on that. I'm going to do a presentation and provide both a little bit of an update on some of the new products that we're developing and some of the new technologies, and then we're going to save plenty of time to do questions. And I think that's a really important part of it. I kind of fill in a few gaps on my story. I did grow up on a farm. That picture was taken 60 years ago, hard to believe. And it goes by in a flash, so grab for every minute of it. I had a chance to be the first in the family to go to college, and after that I spent a postdoc at UCSF in San Francisco. And for me, that was one of the real pivotal parts of my life, because UCSF is where the first recombinant DNA experiment was done. Herb Boyer, Stan Cohen, moved a gene from one bacteria to the next. By the time I got out to UCSF in '78, people had already cloned the human insulin gene, TPA. The pharmaceutical industry was off and running on how it would use GMO technology to create, waves of new drugs. In fact, half of the new drugs that are commercialized today, including all those biologicals for cancer treatment or for arthritis, are all GMO products. My passion was how can we apply these tools to agriculture. And I started looking for places to go to apply and work, and joined Monsanto. In a few years, we were able to introduce genes into plants with agrobacterium, and it's been exciting. We launched our first products in 1996, so literally this spring, we're celebrating the 20th anniversary of the first GMO crops in the marketplace. And it's been fantastic, and excitement of giving me the opportunity to meet a lot of very special and cool people. But I can tell you, it's not just because I'm here at A&M or just because we're at the foot of the Borlaug Institute, but for me, the proudest recognition in my life was being one of the recipients, along with Mary Dell and Mark Van Montague, of the World Food Prize. And it was special for me for two reasons. First of all, you might have heard that GMOs have had a big impact, and at the same time, there's been a little controversy. So, being recognized by the broad international scientific community for the importance of this technology, both took a lot of courage from the World Food Prize, but I think it really, for all the scientists, and I assume a lot of you here, are working in biotechnology or gene editing or GMOs, these are very powerful tools that can help us address, the food security and environmental challenges that we face in agriculture and food production. But the other reason that was really special for me is, the World Food Prize was founded by Borlaug, and after he was the recipient of so many awards and recognition, he formed the World Food Prize to recognize scientists in agriculture. And I had a very special relationship with Norm over the last several years of his life. I first met him in 1990, and we talked a lot about how this technology gets moved into developing countries, what we need to do to sustain that next generation of breeders that will carry on this tradition. And so it's a special privilege to be here today, to be able to do the dialogue and extend the conversation around the importance of technology and innovation in agriculture. Now, when I think about providing a background on where things are, there's enormous challenges in agriculture. You can see from the slide here some of the things that I think are really important. Those of you who are just finishing your undergrad or your graduate's careers, I want to speak to you for just a second, because by the time you're my age, and it's going to go by just like that, the world's going to have 10 billion people. There's 7.4 billion today. We see 10 billion people on the planet. There'll be a large increase in population centers across Asia and Africa. As important as the 10 billion people on the planet, there will be 2 or 3 billion folks who are going to join the middle class as the world economy continues to grow. Their demand, their right to have a balanced, nutritious, protein-rich diet increases the demand for food. So 10 billion people, 2 or 3 billion in the middle class, you do the quick math, the world needs to double its food supply. That means, either through producing more food, wasting less food, we have to double the food supply. To put that in perspective, that means we have to produce more food between now and 2050. 2050 sounds a long way away. It's 34 years. We have to produce more food than we have in the entire history of the world. It's a huge challenge when you think about it. And it's going to be made even more complicated by impacts of global warming. We know that it's global warming and temperature changes are going to change where we grow crops, how we grow crops, some of the challenges in growing crops. In addition to that, there'll be more competition for fresh water, for land, and for other inputs and resources. And finally, we need to make sure, and one of the big challenges is to ensure, as we look around the world, that countries understand the regulatory and policy decisions that they make in terms of the adoption and acceptance of new technologies that are in agriculture and food production. But I want to be absolutely crystal clear. I think we can do that. And I'll talk about all those technologies. I know we have the tools in place to increase crop productivity, to reduce food waste, to achieve that, if science and scientists are allowed to bring these innovations into the marketplace. I mentioned a little bit on global warming, and that's always a conversation where you get into the what's the cause, what's the mitigation. But when you take a look, and this is just a 20-year comparison, you can see College Station. You can see the shift in just the last 20 years of the increase in warming temperature. That's true all around the world. And one of the ways I recognize it is we are now selling corn and soybean seed to grow in Canada. And when I was a kid, you would have never thought Canadians would would be growing corn and soybeans. That's true all around the world. And that's one of the consequences of global warming. But probably in my mind, the part of it that's so important when we think about agriculture is just those small 1 or 2 degree changes in temperature changes when insects hatch, when diseases will break out. And so, the importance of really renewing our focus on understanding the outbreak of pests, the monitoring and the mitigation are really an important part of staying ahead of the impacts of global warming. As I said, I'm an optimist, and I want to be absolutely on the record on this. With the incredible advances that are going on in biology and data science, we will see a transformation of agriculture. If you think about it, these 2 advances are probably the greatest scientific advances of our lifetime. We know the sequence of every single gene in a tomato plant, in a person, in every crop, every weed, every pest. We now have the data science tools that let us map every field in the world, and now are getting to the point where we can take that field, subdivide it literally foot by foot in terms of the changes in soil type, the elevation, and really bring a precision to agriculture in terms of exactly the genetics that we have in that seed and the way we plant that seed and manage it from from an individual farmer's field perspective. We have incredible tools that I think will not only drive that next increase in productivity, but that next green revolution. On the biology side, we've seen incredible evolution in that technology. When you think about early hunter-gatherers selecting and randomly enriching crops, but still doing genetic modification as they made those screens and selections through the advanced breeding techniques and the introduction of traits, and now on the cusp of brand new capabilities with gene editing tools to literally modify every single nucleotide and base pair in that crop. A phenomenal story that I think, as we look forward, gives me both the excitement and the confidence that we can address those challenges of food production. When I give talks, given the association that I've had historically with developing GMOs, or the association that Monsanto's had with crops like Roundup Ready soybeans or Borlaug cotton, everybody expects me to just jump right into the GMO conversation. I will promise you we will get there. But the point I want to make is, as I look back at the last 20 or 30 years, I would say by far the most significant thing that has happened is how these molecular tools have literally changed now the way we breed crops. I remember in some of the last conversations I had with Norm, he would be asking about the drought gene, or how's the disease resistance program going in wheat. And we would talk also about how these molecular sequencing tools and seed chipping tools are letting us breed gene by gene, seed by seed. And it's pretty exciting and pretty cool. I would argue that with these advances that have really occurred in just the last 5 or 6 years, that the way we now can breed crops going forward is fundamentally different than anything we've been able to do in the last several thousand years. The other really important point about it is, with the cost curves for sequencing being reduced literally to less than a penny a data point, with the availability of facile systems for gene transfer and gene editing, these tools are accessible for every crop in every country to move forward with a tremendous acceleration of breeding and crop productivity. In our case, I emphasize, this is what a modern plant breeding lab looks like. The sequencing technology that lets us turn out a genome in a few hours. The seed chipping technology, robots that will pick up an individual cotton seed or wheat seed, shave off a sample to do a DNA test so that you can pick exactly the genetic composition of that individual seed that has the combinations to give you that desired composition of genes. It lets us screen and sort through what would have been impossible. when we first got into the seed businesses in 1997 and 1998, we acquired companies like Holden's and DeKalb. If I would have asked the best DeKalb corn breeder to put together what I think would have been the perfect corn hybrid, these 20 genes, if you just get them together in the same plant, would be the perfect plant. He would have laughed at it, or she would have. Because the odds of doing that in traditional breeding would be 1 in a trillion. And there's about 2 trillion plants that we plant across the US every day, or every year. So we'd have to pick one corn plant in the entire country. Couldn't have been done. But with the markers, the selections, the robotics, you can pick out a 1 in a trillion event in somewhere between 8 and 12 months. That means you can create new combinations that have never existed before that have new potential to drive yield and performance. So the impact on breeding, I think, has been exciting. It's just beginning, and it's broadly accessible across all crops and platforms. Now, I said I'd talk about GMOs. Let me make the point right from the beginning that we live in a GMO world. There's been a huge focus on GMOs in crops. But let me paint a little bit of a picture. I told you the first GMO product ever developed was human insulin. Prior to that, if you were a diabetic, you got your insulin, from the pancreas of the dead animal. The second GMO product produced was chymosin. You make cheese by exposing milk to the chymosin enzyme that cleaves the casein protein that creates solid fat and cheese. Prior to that, you would extract chymosin from the stomachs of young animals. The world's demand for cheese is so big, there aren't enough animals to meet it. And frankly, it's not as good and clean and sterile as producing the protein in the lab. So today, 90% of the cheeses we enjoy in North America, Europe, and the rest of the world are made from GMO recombinant chymosin. Those are just some popular examples. I mentioned half of the drugs launched today are GMOs. A little bit more mundane, but just as important. I'm one of those sloppy laundry guys. I put all my clothes together in the washer and put them on cold, the colors, the whites and everything. The reason I get by with it is I can do it in cold water. The reason I can get by with cold water, all those lipases, proteases, those enzymes in the detergent are all GMO products. So you've taken care of your blue jeans. Of course, all the cotton, is GMO cotton. So we're covered there. And probably my favorite example of these guys, I've got a tank of glowfish in my office. You can go to PetSmart and buy glowfish. Glowfish are tetras and barbs that now contain a gene from either coral or algae that lets them glow under a black light. So you can buy a GMO pet. So, we do live in a GMO world. Now, obviously, in agriculture, in crops, there's been some controversy associated with GMOs. And, for sometimes as a scientist, it's hard for me to both understand and explain that anymore. And here's one perspective that I think about. We have been genetically modifying our foods from the beginning of time. In fact, if you had, come to America with the Pilgrims, the only native foods in the U.S. back then were some strawberries, a few tree nuts, and some sunflowers. Everything else we grow in this country has come from somewhere else. Our corn came from Mexico. Our soybeans came from China. Our fresh fruits and vegetables came from Europe and Asia. They've all been genetically modified and adapted. That's one, we wouldn't be growing any of these crops without genetic modification and adaptation to our environment. And then look at the improvement. I mean, those early tomatoes were little poisonous yellow berries growing on a hillside in Peru. And corn was about the size of my little finger. So we've improved, and that's been a history of genetic modification that now continues even more successfully and more precisely with these breeding techniques. You all know that over the course of crop improvement, we've used mutagenesis, we've used embryo selection, we've used cell fusion to create genetic variability that we then select to produce the better and better crops. Today, we can insert precisely a gene or two using recombinant DNA technology. we can go in and we can mutate every single nucleotide with the DNA editing technology, whether it's CRISPR or zinc fingers or some of the other tools, and that's cool.